So we're going to continue our physiology of muscle by just delving into smooth muscle. So smooth muscle is spindle shaped, and I think you can see the diagrams we have on our slide. Certain things I would like to point out from here is that the actin and myosin are not organized in the usual longitudinal straight microfibril way, and so we don't see any striations when we look under a microscope. But what we can see is this joint, the joining to the dense body. So these brown structures that you can see here. So we have, if we could look at the one that's on the extreme right, what we have are the blue components, which are the thin filaments, and then the pink ones right in the middle, which are the thick filaments. And we see the thin filaments are attached to these dense bodies, and we also see the dense bodies right on the sarcolemma component. So we also observe that there is no troponin, and they have a different form of actin and myosin, which we'll get into details very soon. The dense bodies that are in the cytoplasm are attached to the cell membrane, as I said, and these are the ones that are hold on to, or which the thin filaments bind to. Sarcoplasmic reticulum, they're present, but not as much. They're present, but we don't have as much mitochondria as we have seen in the other other muscles. So we have two types of smooth muscle, one of which is noted as the single unit or unitary smooth muscle, and another one known as the multi-unit smooth muscle. So the single unit, you find that the cells act as one unit, as a sensation, so to speak. So they have these interjoint components among them called the gap junctions. And as we learned in class, we see that when if there's a wave of depolarization, sodium can just flow through these gap junctions right into the next cell, and you find this area of cells acting as though it's one cell or as a syncytium. We see that they are also controlled mainly by non-nervous stimuli, and they are found in hollow organs. On the other end, we have our autonomic neuron controlled smooth muscle, also known as the multi-unit smooth muscle. So each of them is excited by a neuron, or a neuron can stimulate several of them, as seen in this picture, but each of them has this supply from a particular neuron. And the sarcolemma has a thin basement, or a thin membrane that helps insulate or separate the fibers from one another, so that once one is stimulated, it does not necessarily imply that the next one will be stimulated. So they are stimulated independently, and therefore their reaction is also independent by reaction. I mean the mechanical response, which is contraction. So these are noted in components such as the ciliary muscle, the iris muscle, and so on and so forth. Moving on, let's just explain a little bit more about the unitary smooth muscle. And the unitary smooth muscle, if I can just remind you, those are the ones that work as a sensation as one unit. So they have what is known as an unstable resting membrane potential. So it's not necessarily one potential that you see, but it fluctuates towards threshold and then comes back, and so on and so forth. And this, because of this, you see that certain fluctuations actually hit the threshold and there's initiation of action potentials. So they show this continuous irregular set of contractions because of these action potentials that result due to the unstable resting membrane potential. So these are known as slow waves, okay, or basic electric rhythm. And so the maintained state of partial contraction is also known as tone or tonus, and this is what is viewed in these types of muscles. On this slide, I just want to emphasize the fact that the slow wave rhythms are not necessarily action potentials, but these are unstable resting membrane potentials. So they are still at this local unstable potentials. So they determine the rhythm of the smooth muscle contraction and can initiate true action potentials if they hit the threshold, and these are known as spike potentials. So the mechanism there is, once they hit threshold, you find that there's activation of voltage-gated calcium channels, which then allow an influx of calcium around negative 35 millivolts, and this is what causes the further depolarization within the smooth muscle. Let's talk about excitation in the multi-unit smooth muscle. So the changes themselves that lead to contraction are triggered by nervous stimuli, already stated. And the nerve endings are the ones that secrete the neurotransmitters like SEH or noradrenaline, similar to what we learned on the NMJ. The neurotransmitters themselves are the ones that cause this depolarization of the smooth muscle fiber, and the local depolarization is what we see first. Because of how small they are, the local depolarization itself sort of moves throughout the entire smooth muscle and causes the contraction. So this here is very much a distinction that we see from the skeletal muscle. So again, the last point is just there to emphasize that local depolarization is developed because the smooth muscle, the multi-unit smooth muscles, are small, and we don't see really this action potential that is attained. Sort of the local potential is big enough. In our talk about electrical and mechanical coupling, we have been saying the whole point of the electrical activities is to provide calcium so that the mechanical component can take place. For smooth muscle, we see two sources of calcium. We see that the calcium itself can be gotten from the sarcoplasmic reticulum, and we also see that there is an amount of extracellular calcium that can actually enter the smooth muscle via specific calcium channels that are on the membrane of the smooth muscle. Now, one source is not really dependent on another, but we see that the combination of the two makes for a longer lasting contraction. So this diagram is showing the various components or the various ways through which calcium can be introduced. So let's just look at the membrane itself. First of all, here we have the ligand itself can bind to a receptor, and this will activate, so the particular receptor is coupled with a G protein, which can then change GTP to GDP, and this reaction will allow us to change the PIP2, which is found on the membrane, to form IP3. Now, IP3 is the one now that aids in activation of the sarcoplasmic reticulum receptors to then release calcium into the cytosol. The second one is, again, we have a ligand that can bind to a receptor, and this will open up the ligand-gated calcium channels to have an influx of calcium, and again, the cytosolic calcium can go up. Third, we also have a change in voltage that can open voltage-gated calcium channels, causing an influx of calcium, and then again, we have an increment in the cytosolic the cytosol calcium. So we have three ways in which we can identify influx of calcium into the cytosol. All right, now that there is an increase in calcium, what do we have here? First of all, we should note that the myosin is not activated to bind to actin yet, and we need that to happen. So how does this happen? Now that we have increased cytosolic the calcium, it will bind with calmodulin, about 4 calcium bind with calmodulin, and then this complex will activate MLCK. MLCK, this is an enzyme known as myosin light chain kinase. So the active MLCK is going to cleave ATP and phosphorylate the myosin. So once it phosphorylates the myosin, specifically the myosin light chain, which is the one that makes the myosin heads move, we see that it is ready to bind to actin. And once it is ready to bind to actin, binding can then take place, and there's an increase in muscle tension, which is also known as contraction. So let's go through this again step by step. Number one, we have intracellular calcium concentration increasing, and we saw several ways in which this can happen. So it can come from the sarcoplasmic reticulum or through receptors along the membrane itself. Then number two, the calcium will bind to calmodulin. Then we have an activated calmodulin activating myosin light chain kinase. Once that happens, we have here on number 4 the MLCK phosphorylating light chains in the myosin heads, and this increases the myosin ATPase activity, and active myosin then can make cross bridges slide along with the actin and create what is known as muscle tension. So the relaxation is a little bit different from what we saw in the skeletal muscle. First of all, the free calcium in the cytosol will decrease because there's no more stimulation. And then, of course, calcium itself is pumped out of the cell back into the SR, and it can also be pumped out of the cell through other channels that are on the membrane. So the calcium will unbind from the calmodulin. Okay, that's shown here on number 2. And then third, myosin phosphatase is what removes the phosphate from the myosin. Okay, so here now we have to actively remove the phosphate from the myosin. So once this is done, it will decrease this myosin ATPase activity, or the affinity for it to bind to actin. Therefore, you have less myosin ATPase, resulting in decreased muscle tension, or you have less myosin actin activity, and thus you have a decrease in muscle tension, and this is known as relaxation. So far, we just want to appreciate some of the differences in what we have learned. The striations, we see them in the skeletal muscle as well as cardiac muscle, but not in either of the smooth muscles, while actin and myosin are noted in all four. The level of control, as we had introduced, is voluntary only in the skeletal muscle, and the neural input is somatic, meaning voluntary, of course, in the skeletal muscle, while in the others we see an autonomic, more automatic. There is a neuroeffector junction, which is noted as the neuromuscular junction, very specific in the skeletal muscle, and then we have others that are diffuse, noted in the other types of muscles. In terms of hormonal control, none is noted in the skeletal muscle, while we have several depending on the locations in both types of smooth muscle, and epinephrine very observable in the cardiac muscle. How about the sources of calcium? While in the skeletal muscles we only see this happening in the sarcoplasmic reticulum, the ECF is also a source for all the other three muscles. Other three components are the fact that the regulatory proteins that bind the calcium are really troponin in skeletal muscle and cardiac, whilst in the smooth muscle we depend on calmodulin. Gap junctions are observed in the smooth muscles, specifically the single unit ones, and also in cardiac muscle, but not in the skeletal muscle or the multi-unit ones. Pacemaker activity, meaning those that sort of reach threshold on their own without stimulation, that is not observed in skeletal muscle or the multi-unit smooth muscle, but we see this in the single unit as well as some of the cardiac muscle cells. So for now, or as well as the cardiac muscle cells, let me holistically put that. For now, we'll end here.